Okay, thanks.
Thank you. Yeah, so, some of the low hanging fruit here are, we have candidate types of cells or tissues that we would want to target. And then we also have potential markers to target. So I feel like this could be one easy way to collaborate, because we could have like a solid plan, this is what we want to do, and set something up.
What would be the kind of cells to target first? What would be the low hanging fruit? If you had a way to target specific cells, what would you go for first?
Yeah. Well, I think senescent cells would be really nice. Put this in BioTool to the test on the senescent cells. So for example, in my lab, we do the surfaceome characterization of those. So we have cell surface targets on cell type specific senescent cells. So, if it's a senescent monocyte or a senescent fibroblast or pre-adipocyte, right? Then we can target those specifically. And I think that'll add an element of precision to the senolytic intervention. Now the question is, what is the way that you would target those? What are the tools that are available?
I heard synthetic receptors have a big potential there, designing synthetic receptors that bind to those surface markers and using those synthetic receptors to guide delivery cells as delivery vehicles to carry cargo to those specific types of cells.
Oh, cool. So do those work by you introduce the synthetic receptors in like the senescent cells? For example,
No,
or-
in the cell that is carrying your senolytic drug or your therapeutic molecule. So
Oh, I see.
yeah, you like load your drug molecule into a cell, give it a specific receptor, or give it a synthetic receptor that will target the senescent cells. And that should, in principle, drive the delivery of your senolytic drugs to the senescent cells with those markers.
Okay, yeah, that would be straightforward. Is this something that's been, now you guys have tested in some,
A little
system?
bit. I'm not the receptor guy in our lab, but I know that people are putting together like chimeric receptors similar to CAR T receptors, just targeting different biomarkers, and using those in hematopoietic stem cells, and differentiating those into platelets that have those kinds of receptors on them to specifically target cells that have those surface receptors, or
Okay.
cells that have the biomarkers.
So these are platelets that you're engineering that deliver the payload of
Yeah.
interest. Oh, interesting. Okay. And does this have, is it, preferred in some kind of tissues versus others? You guys have done this like in a mouse or,
No.
okay, in cell
It's
culture
definitely
it
cell
works?
culture. Yeah,
Okay.
so just delivering from platelets to a reporter line like HEK.
What's the advantage of platelets compared to other delivery vehicles like exosomes or,
Exosomes and like extracellular vesicles still have like a big draw. The advantage of platelets is that they're anuclear, so are exosomes, so you can radiate them for like sterility purposes. They're also naturally able to target certain tissues, like they naturally like co-metastasizing cancer cells. They naturally home into like areas of inflammation and injury. And then they also just tend to be carrier cells, and how they degranulate and just kind of like dump their contents on things. But they're not the only way to do it, for sure.
I guess they're the body's natural way of doing this.
Yeah.
Right. How easy are they to engineer?
They're kind of tricky. The production of them is a bit labor intensive because the megakaryocytes that produce them aren't self-replicating, so you have to go from megakaryocyte progenitors and then control their differentiation into producing platelets. And then for getting the platelets out of the megakaryocytes themselves, there are a couple different techniques for like passively kind of shearing them off of the MKs or trying to activate the MKs and releasing them. So they're labor intensive. Sure.
And what kind of cargoes do you load them with, in what you like with something toxic or like what?
That's what I want to get to with my dissertation project is gonna be loading, platelets with, split Intian toxin, so like split portions of a toxin. But so far we've done, Cree recombinase. We have a reporter line that just changes from green to red once it gets Cree recombinase. It like cuts out the green part and allows the red part to be expressed. So we've delivered whole Cree recombinase and then also like split Cree recombinase. But we haven't.
So it's like reporting on that it's successfully
Reporting is,
delivered. Yeah.
but I at least haven't gotten any further than that yet.
Okay. Oh, that's interesting. Yeah. So this could be like an early thing to try. I mean, because you're already doing this in cell culture, right? So I mean, an easy thing and relatively simple thing to try then as like a proof of principle would be to, make a induced senescent cell culture and then deliver some platelets to those, right? With something that hopefully is more toxic to senescent cells. So, I mean, the thing is, depending on how targeted and specific the delivery is in vivo will be probably depend on what you load. Because if it's very specific to just senescent cells, then you could make something more toxic. But if it's kind of might hit some healthy cells, then you want to load it with something that's like preferentially like senolytic. So would meaning that it's more toxic to senescent cells versus non-senescent cells, and there are some candidate things, that could be used. So I think like, for example, there's the some senolytic peptides, like the oxopeptide, and then there's the I don't know what you can load. Can you load drugs into these or compounds or?
So far, we've just done the products of genetic circuits, so something that we can have a gene encode the megakaryocytes or platelets to make. So I'm not sure about it.
Okay. Yeah. So we need something that could be genetically encoded to be loaded in. Okay.
I could be wrong. Tara would definitely know more, but that's at least what I've done so far.
Okay. Yeah, that would be interesting. So.
I guess the targeting efficiency would be as good as your marker for the senescent cell. I know for CAR T, they do have some off-target effects. So what they're doing is loading two receptors on the CAR T to recognize two different markers on the cancer cell. So it's a little bit more refined and targeted. So
yes, and CAR T cell would also be an interesting option, I think, in this, And so what is those cells just kill cells, right? So they.
I'm not an expert on CAR T, but they do trigger some sort of cascade in the cancer cell that leads to their death. I don't know if it's apoptosis, it's probably apoptosis. But it's specific, again, to the cancer cells based on the cell surface markers that they've identified. But you also kill normal cells in the process, so that's why they're trying combination receptors.
Okay. Yeah. So I mean, this cell culture experiment would be something that would be relatively, straightforward to try. Maybe we should connect with Tara also. See,
I definitely.
You want to do this in your lab? Well, we can always connect also later, so you guys feel free also we could follow up after the meeting or, and discuss it. So,
definitely need to run it by her before.
Of course, yes.
I'm a grad student, I can't,
Yes, yes, yes. Oh, yeah, I understand. Yeah, totally.
but just for brainstorming and such, I think it could potentially be a pretty cool avenue to look at.
Yes, yeah. Yeah, I'm thinking, what are the other targets that could be used? Are these for delivery? Is it always something like a cell surface target, or is there other basis for targeting delivery? Could it be a secreted or,
It maybe could be secreted if it stays relatively localized. My concern would be if it gets secreted and ends up like endocrine system, then.
yeah, ends up on another cell?
Right,
Or I wonder, like, is there some, how in nature, a lot of things follow gradients, so it gets
Oh,
more
yeah.
concentrated as you're closer. I don't know if there's like synbio people exploit that mechanism in some
I
way,
think some
or,
people do,
Uh-huh.
just like chemotaxing
Yes.
towards certain signals. There definitely are cells that do that, but I am not as familiar, but
Mm.
that could be an avenue to
Yeah,
look at.
okay,
I mean, I guess fibrosis could be sort of another target for you to combine. Just thinking of chemotaxis, not that I'm an expert in this,
Mhm.
but just combining it with something else or an environmental cue for aging.
Mhm. Yeah, that's a
may be able
great
to
idea, yeah.
target. Like, I don't know how the microenvironment is around senescent cells or tissues or aging cells or tissues, but I think cancer people are starting to target the tumor microenvironment more than the cancer cells. I think we heard something about it
Mhm.
during one of the talks yesterday. So maybe thinking of it as the cells in their environment and targeting both could be an avenue.
Yes,
I don't know. how that
that's
would be,
that's a really cool idea, I think. I think in aging, it's maybe not as well of a developed, aging microenvironments isn't a as developed a concept, but I think it's really true. And for example, we do know a lot of, actually, like, your fibrosis is kind of happening everywhere as you age a little bit, kind of how Jennifer had said yesterday. And the stiffness of your organs actually increases with you as you age, but I think yeah, if there's some way, part of that is that your ECM is getting damaged, and the damage to your ECM often is things that just don't get repaired. These are very slowly turning over proteins. They accumulate modification, so it's not something easy to repair. But if there's a way to deliver to the ECM damage, or something like that, could be interesting.
With delivery to ECM, I know that there are some relatively common targeting motifs or like synthetic peptides that reproduce the common motifs in ECMs to attract cells to them.
Mhm.
So I'm wondering if we might be able to use something along those lines for targeting the ECM.
Okay, so you can use some of the sequences in the
Yeah, for like targeting to fibrin or laminin.
Okay.
Like the RGD peptide is a common one. That is. Just had to memorize this for my quals. And it's
So
already.
if there was a way to use the RGD peptide or something else to take the cells or the cargo, say the platelets over there, and then have a specific receptor on the platelets to recognize the senescent cell when they're sort of in proximity, and then
Mhm.
specifically deliver the cargo to those cells.
Hmm.
Could be like a two-step, a combined two-step process. Could be.
Yes. So. Yeah, so one to attract cells to the microenvironment, and then one to target the cells in the
in that
microenvironment.
So could I jump in? It's okay, quick
Yes.
comment. Just thinking about the environment or the microenvironment and fibrosis, what about thinking generally about inflammation? and like
Mhm.
inflammasome related aging or inflammaging. So just inflammation in the microenvironment. I don't know, like, so maybe targeting specific inflammasome activation, things like that
Yes.
would be an idea.
Yeah, that's
And
true.
the whole environment came up too, and particularly in the brain. I think this came up in one of the breakout rooms I was in yesterday, that you want to think about target, about neuronal survival, et cetera. You don't necessarily think about like specifically doing anything that targets the neurons, but that if you thought about astrocytes or microglia, and those, and particularly microglia in terms of creating inflammation in the nervous system, but obviously that would be applicable to macrophages and the periphery, et cetera. That might be something to target. The increase in inflammation that happens with aging.
Yeah, that's a good point. Yeah, so targeting to the inflamed or fibrotic environment are both interesting ways to do it, I think. And then I think in the and then I guess once you target that, what do you target? You target it, and then what do you deliver? I guess is the
Yeah.
question also in that case.
But I mean, maybe generally just understanding. I mean, this is maybe focused on delivery, but I think there's a lot to just understand about how the process changes with aging, right? How does inflammation and fibrosis, so I think maybe identifying, maybe in an organoid context or a tissue context, where that's happening, right? Because it's useful to understand the pathways on a cellular level, right? But it's maybe not happening the same. So I think it's not a targeting, like, okay, get rid of it, but just there's some discovery, I think, that could be applicable.
Yes.
Using reporters or things like that, or I don't know. There could also be just kind of like some kind of, I don't know if you think about general screening for senolytics. I know there are specific, maybe things that you can think about targeting, but I don't know, maybe there's some discovery aspect. That's not necessarily maybe the greatest thing for grants, I don't know, but I think just brainstorming, right? What you could get out of a system that identifies changes that are associated with the aging process.
Yeah. Yeah, that's also, that's interesting too, because we always hear about inflammaging, of course. I'm sure you've heard that a million times, Raquel, too. But it's a very general thing, and we haven't really done very like precise quantification of how this is happening during aging in vivo. So yeah, like you mentioned the reporter. So what if you could have a reporter and follow this over time? Where is the inflammation happening? And
yeah,
when
that would be very cool. Mm-hmm.
Yeah. So in vivo reporter. And then is it possible then, if you were delivering with this is kind of the sentinel cell idea, actually, right? Is that so? I wasn't sure what that even meant, but they mentioned was one of the breakout room sentinel cells. So like if you have like say these platelets or other cells going to the location and now acting as reporters in some way, is that a sentinel cell idea?
That was my interpretation of it, was just something you can administer systemically, and then it will kind of recognize these areas of aging inflammation and then report back.
Okay, yeah. Yeah, in that case, even different aspects of inflammation, or even like you could detect things like DAMPs, damage associated molecular patterns or pathogen associated. So places that have things associated with pathogen, maybe that's a type of inflammation, or DAMPs, which are more like general wounds. So if you have blunt trauma or something breaks the cell, that's another thing, DAMPs are released, and that's another source of inflammation. So kind of tracking these different sources of inflammation down in vivo could be interesting.
And then they could detect different kinds of inflammation, but only report back on the age-related fibrosis, for example, something like that. It could be a sentinel that sort of surveys everything, but then only reports back what you're interested in. So you can further engineer it for other purposes, but I think it's good to have an idea of what's going on, kind of
Yes.
information are around.
That does sound difficult to get that kind of specificity, but it
Yeah.
does sound like a goal to aim for.
Yeah. So, what about in terms of delivery, are there, is there capabilities for cell type specific delivery? Are there capabilities for delivery in, like the brain, for example, for interventions?
Brain's hard.
What a brain area.
That guy,
Yes.
that guy's a stickler.
Does his job well.
Does his job well.
Mm,
For tissue or like cell type specific, I think as long as we have enough selective biomarkers, we could probably engineer cells with specific enough receptors to be able to target delivery to specific types of cells. I think bacteria as a delivery vehicle also have kind of been interesting to me. More in like I'm more familiar with the cancer aspect of that though, with just like targeting to the tumor microenvironment, like Foley's toxins and whatnot, but there's a lot of synthetic biology that we can do with bacteria. So we can have some pretty interesting, like optogenetic and very controllable circuits in bacteria for delivery. And I don't know if that has any potential with aging, because bacteria doesn't home into like aging as much as it does with tumors, but I think that could also be an interesting alternative to platelets.
If there's anything soluble in the microenvironment that the bacteria can sense, you could also use it for that.
Potentially,
Like nutrients or
but
any other inflammatory in a molecule that's sort of soluble outside the cell.
yeah,
You could engineer bacteria to sense that.
oh,
and then
Yeah.
report back.
so this is like gut bacteria that you're talking, or?
They usually, you're the expert, that's fine.
E. coli and different types of strep
Mm,
are often engineered, but they're specifically developed to be oxytrophic to things that are only found in like the tumor microenvironment. So like it's a different nutrient profile, I think is the right word. So you can engineer the bacteria such that they only survive with the nutrients that would be in the tumor microenvironment. So I don't know if aging microenvironment would have things that bacteria would need to survive that can help with off-target effects, but I don't know if that's
yeah,
um
I don't know if we have quite that level of specificity, or like, maybe aging is a little bit more subtle than the tumor microenvironment,
Yeah.
so it would maybe hard to distinguish.
yeah, bacteria may
Yes.
not be a feasible pathway. I really like how they work in cancer, so I want to keep them just kind of on my mind.
So. And what about other modes of delivery or payload delivery? So, I guess there's, virus and there's exosomes, things like that,
Yeah.
Particles.
nanoparticles. So are those all, things that can be equally engineered? I mean, engineered equally as well and targeted equally as well as,
I'm not familiar.
yeah.
I don't
I'm
know.
not super familiar either. I know people are working on them, but I don't know if one's better than the other.
Yeah. I think it's something to keep in mind for sure, but I don't think I can speak very confidently about them.
Right, yeah. Be interesting even to, yeah, maybe Actually, this actually goes into sentinel cells too, but thinking of being a reporter, yeah, because it's nice when you make something into like now a biomarker or a signal that you can measure for that aging hallmark. So something that measures the inflammation or and then maybe secretes or reports so that you can measure it when you're actually using interventions too, or you know, it could be really interesting. So yeah. Are there anybody in the Zoom have thoughts they'd like to share? We have one in the chat box that somebody.
I don't see anything in the chat. The chat is just talking about the captions. There are captions that are being generated. So just a couple of things that popped into my mind about like delivery. I know people, I think maybe related to exosomes, but maybe more controllable, or like liposomes, things that sort of can also take advantage of cellular endocytosis and release. I know that the people that are studying sort of like, you know, how cells normally take up things like liposomes and then release them in a non like activating way, like not to not get to liposomes, to not to get to, I'm blanking where lysosomes are. You know, to not target for degradation, right? That would to uptake, you know, sort of you know, these, you know, membrane sort of vesicles and release things. So it would be another thing. And then I know that I've come across in reading like hydrogels that can maybe, you know, be used maybe for like local delivery, right? In vivo, like that you could, you know, either have drugs or some of these delivery particles, right? So that you could like deliver them locally, right? Rather than maybe something that's systemic or just, you know what I mean, in tissues. So those are just some kind of brainstorming things to throw out there.
So these hydrogels are things like you can introduce, you inject or surgically implant it like a.
I think
material.
so. I think that's the idea, right? That would give you like a localized. So let's say maybe, again, I'm just brainstorming, right? But you have sort of, you identify areas, right, where aging is happening, right, where there's excessive inflammation, fibrosis, whatever, and you know, maybe you need to target that area, right? How do you, you know, you identify it and you target area if that's amenable at all, you know what I mean, with to and then something that's injectable, you know, to deliver a material in a localized way. Was maybe an idea. Again, I'm just totally brainstorming.
Yeah, there are some of it is microfolio as well. There are biomaterials where they make these scaffolds and they combine it with cells and implant them, you know, under the skin or, you know, wherever you want them. So the cells are making drugs or releasing products within that biomaterial that's implanted over a determined period of time.
Okay.
So you could do that. So you could engineer your cells and then integrate them with the scaffold, and then put it where you want it to go. So there are ways of doing that.
That would be one way to do the brain, because I know that they'll, like after taking out glioblastoma tumors, they'll put like wafers with with drug in them that like degrade over time and release drugs. I don't know how bonkers it would be to have cells producing a drug for that. That might have been.
The immune. I mean, I guess brain is a little different, but the issue with that would be the immune response to the cells within the scaffold, right? But you could also modify biomaterials and covalently link them to certain drugs or, you know, compounds that are released over time, and you can sort of play around with the extended release times, let's put it that way. But that's more like in vivo.
Yes.
But that's
It's
that would be
interesting
one
though how
way.
you can, so you can kind of time, you can engineer how long the intervention is applied though, these kind of things. That's interesting.
Yeah, you could do that.
So, well, you know, I think besides, I know this is the drug delivery session, but besides, because I do, my brain, when I think about things that are like, what do we have some data on right now that can be the source of collaborations also, you know, for example, we know other things about senescent cells or things that we could kind of exploit. We understand a little bit the mechanisms and their markers that are increasing or triggering senescence. So if there can be some kind of synthetic circuits built in that respond to those cues, you know, in senescence, it would be interesting to probe, you know, just the biology of senescent cells and probe for like, what are their weaknesses as well, potentially.
I have two basic questions. How long do senescent cells sort of hang around? And are there stages to senescence? Like a cell has to go through certain events, I guess, to be fully senescent, I'm assuming.
Yeah,
that
it is,
the
there
thing?
are stages, like once they're committed, I think they're in,
Okay,
but they change over time. So, you know, it's an evolving phenotype and they're dynamic, they respond. So if their environment changes, they also will change. You know, they don't start to divide again, but they secrete and their phenotype can still change.
okay.
They're very metabolically active.
And they're the good
and-
versions and bad versions too.
Yes.
So,
And the thing is, the senescent cells, they're, you know, the idea is, I mean, I think one of the main thoughts in the field is that they're supposed to be transient. So they have a role when they're, like, say you have a wound, then senescent cells will be there around where you had that trauma that were induced. And then they actually help to resolve the wound. They secrete things that help resolve the wound, and then they just die. But with aging, for one reason or another, senescent cells tend to accumulate more in all your
Okay.
tissues. And then this persistence of these cells is what, and their secretions is what drives bad phenotypes.
okay.
So, yeah, if there were ways to like limit the duration of senescent cells, that would also be cool. Like maybe that's a strategy to allow them to do their job, but then get rid of them, you know, something like that. Be interesting, I think, thing. Yeah.
Self-destroy after your job is done.
Yeah.
Oh, that's interesting. I wonder if there would be ways to target specifically for senescent cells that have kind of overstayed their welcome. I don't know if they signal any different
Yeah.
or how to differentiate them. But
You know, that would be interesting. Yeah, people should look. In culture, we can culture them different lengths of time and like if they're SASS, it changes a lot. I mean, it's crazy. It changes a lot. And I think the early few days are completely different from like day 7 to 10. And then I think it continues to change after that. But I think these early few days is probably the critical period that you want to keep around. So targeting things, yeah, early versus late. I think that would be interesting. Yeah.
I
Let's
mean,
go.
would you come up with a reporter that marks that transition from early to late?
Yeah,
With
That
some
would be
gene
cool.
circuits.
yeah. Oh, yeah. I like that idea.
even be something like, when they transition to late stage. You have the reporter that marks it with the expression of a specific cell surface receptor that's nowhere else. And
Yeah,
they can target that externally to kill those cells or study those cells. or-
Yes. yeah.
So
And
those
it
are
would
the things
actually
that
be
seem like-
very interesting, just from the reporter perspective. When we look at accumulation in vivo with aging, is it, if we have a reporter for early versus late, is it really just like the late ones that are accumulating?
Right,
Yes.
that you don't even know that.
Yeah, because we don't have I mean, that's the idea, but we don't have a reporter that good. We have senescence reporters, but not an early versus late
Mhm.
distinguishing, one. So even
Yeah.
something like that would be interesting to probe. Yeah,
I think if we could get biomarkers that were distinct enough between early and late senescence, I think we definitely have the tools with SynBio to come up with a reporter cell.
Mm.
Uh,
yeah, oh, that would be interesting. Okay.
So again, a very naive, but maybe. Do each cell type in the body have their own version of senescence, like that? Do macrophages go into senescent macrophages, or like, are there, am I making sense?
Mhm.
Do these different types all have their own versions of their senescent self?
Yes, that's a good question. I mean, they all have, presumably they all have senescence. And the phenotype is different for each, to some extent. So probably, but honestly, the field has not gotten that rigorous yet. But probably with like the SendNet Common Fund initiative that they have, that this is one of the things they'll figure out.
So when you have those like senescent cells in a dish, what kind of cells are you starting out with typically, when you
Yes.
use senescence? Is that
Most people look at fibroblasts.
fibroblasts?
Yeah, that's where it's the most well defined, so many people have started there.
Mhm.
And actually, that's a little bit of the limitation of what we know about them, is that a lot of our markers are based off of the senescent fibroblasts. But then as people look in more and more cells, they realize they're not the same,
Mhm.
right? So it's what we learned in the fibroblasts isn't necessarily true for these other cell types,
I did a little bit of work with retinal pigmented epithelial, just like RPE, kind of common model cell, but we were using senescence associated beta gal as a report for quiescence, actually.
Oh, for quiescence?
Yeah.
Oh, really? And it worked?
It does show up a little bit with
Oh,
quiescence. even if
Oh,
we're not-
no.
So,
Yeah, that's why our markers are not so good for
Right.
senescence, right? Yeah,
um, oh,
yeah, RPE cells though. Yeah. Yeah, so this is why in the senescence field, we often have to do like combinations of markers. Actually, that's what makes it so tough. But the one thing we know is that the reason we know senescent cells are involved in aging is because of these transgenic mouse models driven by P16. So under the expression of P16, they report senescent cells, but under the P16 promoter, you can selectively eliminate senescent cells. So you give them a compound, and that for cells that express P16, it activates the apoptosis program. So that's how we know, because they did this in these mice, they naturally age them and they would just eliminate senescent cells once a week, and they live longer, all of their age-related diseases. And many labs have now studied these with these mice in many diseases. And that's how we know so well, exhaustively, that senescent cell elimination is a promising therapeutic target, at least just in mice, and people try to do it in humans. So in that sense, we know P16 cells are important, even if there's not one specific senescence marker we know at least from those studies that the P16 cells eliminating those is good. Right, so in a way, the P16 is a nice,
So that's
marker.
the starting point.
yeah.
What does P16 do?
It's like a cell cycle,
Yeah,
Yeah, regulator, so when it activates, it'll help out, be part of the cell cycle arrest program for this.
So cycle arrest.
But the thing is, it's funny you brought up macrophages, because those are one of the cell types that express P16 when they're not senescent in some contexts. So, there's some thoughts in the field that like these aging effects are also from elimination of not just P16 senescence, but P16 macrophages could have also explained maybe part of the anti-aging phenotype of those mice. Yeah.
That would make sense. And macrophages, inflammation, tie up together, right?
Yeah.
People also use macrophage derived extracellular vesicles for delivery. By the way,
True.
they work much better than actually using macrophages themselves to deliver things. But they sort of engineer the macrophages to produce whatever it is that they want to deliver. And then they naturally assemble them into extracellular vesicles that shed, and we just collect them in the media and then use that. Another method.
Yeah, that's nice. The macrophage.
Derived extracellular vesicles.
People also will apply CAR, like the chimeric antigen receptor stuff to macrophages. So instead of doing CAR T, they'll do like CAR macrophages or CAR neutrophils,
Oh,
which
really?
is kind of interesting. So instead of relying on the cytotoxic T cell, they rely on the macrophage's ability to eat things. But I think it's neat how much people are expanding the macrophages. I think they're a very fun cell.
Yeah. I mean, in theory, you can reprogram any kind of cell, right?
True.
So, it's great that people are expanding from T cells into macrophages and other things. Another sort of basic question. Do people in the aging field, and I heard Nam talk about the systems, biology aspect of it. Do people in the aging field or systems biology of aging think of it in terms of gene regulatory networks at all at that level, or it's more signaling pathways level? What kind of system level are we talking about there?
I think so. Not me, I mean, I don't think about it that deeply in that level, but I think that there are definitely people who do think about the gene regulatory networks that are changed, during aging and, but really that's not my specialty, I'm not sure.
But that's also another potential area of bringing in synbio, because you can do synthetic gene regulatory networks as well. To study processes. So, I meant to talk not about it, but.
His paper was very cool that switches between the two cell states. He did one in yeast, he made yeast replicative lifespan extended longer than ever before, I think, he would just he could toggle them between two different, like they called aging states in the yeast. So he would, by oscillating back and forth between the two aging states, the yeast lived longer, much longer,
Sounds like everybody needs a reset. Every once in a while.
Yeah.
Can we toggle our different aging states? Trying to think of what are different other needs in terms of targeting in the age, in aging. I think this is a very general thing in diseases also. I mean, we happen to have one type of cell, that's senescent cells that we know that we're targeting and killing or intervening in some way. Are there other things in aging specific? I wonder.
I wonder about the eyes a little bit. I don't think I've heard folks talk about eye disease, like cataracts and stuff. Wonder if that's an area of delivery that should have more focus. Also menopause. Carmela brought that up in the like pre-meeting thing. And I thought that was pretty interesting to think about. Just that might just be more of a hormonal therapy sort of thing, but
Yeah, that's an interesting aspect of aging that really has not been studied nearly enough. It's so surprising. I guess not surprising, but also just really shocking how little it's been actually studied, and it's the fastest, the reproductive aging is like, it's happening faster than all the other organs.
right.
And we don't, they kind of studied it that much. So, yeah.
Oh, I remember my question now. So there are cases presumably with genetic underlying causes where aging is accelerated, like that boy, like, who looks like an 80-year-old guy. Have people studied those cases? Like, is there, I can't imagine there's like one silver bullet of this gene or anything, but like, has anything come out of those cases where there is potentially a strong genetic component and what drives aging in those cases at all?
Yes, I think the one you're thinking of is probably Hutchinson-Gilford progeria, where they're very young. Yeah, and they look very old at the very young age.
But are they really old? Like, or do they just look old, or are they,
Yeah, that's a good
biologically
question.
old?
That's a good question. So they reproduce, every progeria is what they call segmental, which means it recapitulates some aspects of aging, but not others. And the progeria like that just happens to recapitulate multiple aspects of aging and look a little bit like aging too visually, right? But I think they're useful and people have used them, and oftentimes things that are good aging interventions also work in those, to extend lifespan in those models. So they are, I think, like potentially good for screening interventions, right? Something that works in those models might also be good for, normal aging. But since a lot of them have a known genetic mutation, you can kind of track the mechanism. They can kind of track the mechanism down to that one thing, it's a specific mechanism. But yeah, it's interesting that sometimes this one thing can produce so many widespread defects in the cell that mimic aging in a way. But I think, for example, the progerin is one of these proteins that kind of accumulates in the nucleus and kind of causes disorganization of chromatin and stuff. This is one of the things that changes a lot in one of the progerias, but then they would go and find, during natural aging, this is actually going up too, just to a much lower extent. So, maybe, in some cases there could be a link to a real like natural aging mechanism. So,
Yeah, I always thought it was very interesting.
Yeah, these are interesting cases. And I mean, like, even, people look at high fat diet as an accelerated aging too, because it does it is segmental, but it causes like your brain aging to go faster, and of course the metabolic aging to go faster. So multiple aspects of aging are accelerated in those models too. Yeah.